my final module for Module 3 by Lindsay Cox for Brightpoint Community College. This is Samantha. She will be interrupting. I can't tell when, I can't tell why, but she loves it whenever I have a camera on, so fair warning. We have cell cycle and mitosis. 1. Describe a function of a chromosome carries genetic information needed for growth, development, and function of an organism. Then I have my picture here, the organism. 2. Describe a parts of a chromosome. Centromere hold sister chromads together, telomeres protect ends, and chromads carry identical DNA copies. As you can see, there are the chromosomes right over there. Slide 3. Describe the difference between autosomes, sex chromosomes. Autosomes are non-sex chromosomes, and sex chromosomes determine biological sex X and Y. We have the autosomes and all the chromosomes over there. 4. Define haploid and diploid karyotypes. Haploid is equals one set of chromosomes, diploid 2 sets, one from each parent. You can see the pictures over there. 5. Demonstrate how you can read a karyotype indicating the sex of an individual. Look for X and Y, XX, female, XY equals male. Then there is the whole Punnett square as my picture. 6. Describe the difference between interphase M and mitotic phase. Interphase is equals cell growth and DNA copy. M phase equals cell division. 7. Describe how the cell changes during G1 phase. Cell grows, performs normal functions, prepares for DNA replicas. So you can see there's all those right over there. 8. Describe how the cell changes during the S phase. DNA is replicated, forming identical sister chromatoids. Describe how the cell changes during the G2 phase. Final growth. Cell checks for errors before mitosis. Then there's the picture of the cycle right over there. Describe how the chromosomes and cell changes during the prosphase phase. Chromosomes condense, spindle fibers, nucleus starts to break down, and then there's the prosphase over there. Slide 11, describe how the chromosomes and cell changes during promophase and nuclear envelopes disappear, spindle fibers attached to chromosomes. You can see the phosphate over there. That's my picture. Describe how the chromosome cell changes during the metaphase. Chromosomes line up in the center of the cell. 13. Describe how the chromosomes and cell changes during the antiphase phase. Sister chromatids separate and move to the opposite side, so you can see metaphase 1 and anaphase 1 as well. 14. Describe how the chromosomes and cell changes during the telophase phase. 2 nuclei form, chromosomes loosen, spindle disappears. You can see all of those chromosomes happening over there. Slide 15, describe a situation in which a cell might enter a GO phase and why it's important. Resting phase, some cells like neutrons stay here permanently. See, there's the resting phase over there. Section 2, mitosis. 16, describe why crossing over during mitosis is important to humans. Crossing over increases genetic variation by swapping DNA between the homologi and chromosomes. 17. Describe how the chromosomes and cell changes during the prosphase of 1 of mitosis. Homolysis in chromosomes pair and crossover nucleus envelopes breaks down, and you can see it, all the chromosomes breaking down over there. 18. Describe the chromosomes and cell changes during the metaphase 1 of mitosis. Homoiglous chromosome pairs line up along the middle of the cell, and pairs of homoiglous chromosomes move to the equator of the cell. 19. Describe how the chromosomes and cell changes during the anaphase of one meiosis. Homoiglous chromosomes separate and move to the opposite sides. As you can see, there is all of the cells of the chromosomes breaking down. 20. Describe how the chromosomes and cell changes during the telophase of one meiosis. Two nuclei form, each has half of the chromosome numbers. And you can see it coming together and then it splitting apart. 21. Describe the differences between meiosis and meiosis. Mitosis is two identical cells. Meiosis is four genetically unique gametes. You can see it all separating there. 22. Define deletions, duplications, inversions, and translocations in chromosomes. Deletions, duplications, inversions, and translocations are chromosome structure errors. You can see it all breaking down over there. 23. Describe the difference between aneuploidy and polyploidy. Aneuploidy equals mixing extra chromosome. Polyploidy is extra full sets of chromosomes, and you can see all the sets of chromosomes on the slide. Section 3. Mendelian and inheritance. 24. Describe the difference between a trait and a character. A character is a feature, a trait is a version of that feature. And there is your genetics vocabulary as my picture. Slide 25. Describe the difference between true breeding and hybridization. True breeding are same traits passed down. Hybridizations cross between different traits. And you can see it all breaking down over here between the difference. Sorry, my cat is doing her thing, like I said before at the beginning. So if you hear sounds, that's her. 26. Describe the difference between self-fertilization and cross-fertilization. Self equals same point, same plant. Cross equals different plants. 27. Define the P, the F1, and the F2 generation. P equals parents, F1 equals first kid, F2 equals grandkids. And you can see the generations here. 28. Define genes. Genes are DNA sections that code proteins or traits. And then there's all the lovely DNA sections I created. 29. Define alleles. Different forms of a gene. And you can see in the picture that there's all these different alleles and their types. 30. Define dominant genes. Give an example. Dominant alleles show even with one copy. Example, brown eyes. 31. Define recessive genes. Give an example. Recessive genes, recessive alleles need two copies. Example, blue eyes. Define monohybrid cross. A genetic cross studying one trait. And you can see that there's all these combinations over here. 33. Demonstrate how to solve a monohybrid cross using a Punnett square. I have T, big T little t times T, big T little t equals TT, T little t, and then two little t's. Define dihybrid cross. Cross defining two traits at once. And then there's a little bunny. And then all the variations that can happen with your genetics. 35. Define how to solve a Dolly hybrid cross using a Punnett square. Big R little r y, big Y little y times big R little r. Big Y little y equals 9:3:3:1 ratio. So those are all the combinations. 36. Describe a pedigree. That is a family tree showing inheritance patterns. Provide 37. Provide an example of a pedigree. Identify each individual using proper symbols. So circles equals female, squares equals male, shaded equal affected. 38. Describe the difference between wild type and mutant alleles. Wild type equals common form, mutant equals change version, and you can see all the versions here on what can happen. 39. Describe sex-linked genes. Genes found on sex chromosomes, usually X. And there's all the pictures of chromosomes. 40. Demonstrate how to give a sex-linked gene problem using a Punnett square. So X and X times X and Y equals 50% carrier, 50% affected sons. There's just a gene right there. 41. Define the difference between sex-linked genes and sex-influenced genes. Linked equals on XY influence expressed differently in sexes. And then there's a Punnett square where there's very many different variations. 42. Describe an incomplete dominance. Neither allele is fully dominant blended traits. And then there's all the alleles over here and dominate. Demonstrate how to solve incomplete dominance using a Punnett square. R equals R equals R pink. And there's a Punnett square of the flowers and all that fun stuff. 44. Describe a commutance. Both alleles show equally. And you can see there's heterozygous and homozygous. 45. Demonstrate how to solve a problem using a Punnett square. CR times CW, CR, 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 CW, CW, CW. And there's the picture of the Punnett square. 46. An ABO blood type is typing an example of a codominance in multiple alleles. Inheritance. Describe the genotype of each blood. So A equals double A, AO, B equals double B, BO, AB equals ABO equals double O. 47. Demonstrate how to solve a paternity problem using ABO blood type using a Punnett square. O child equals both parents must carry an O allele. And how that shows how the alleles can go down. Define gene linkage. Give an example. Linked genes stay together unless crossing over happens. So there's all the genes and how some of them cross over there. Section 4, non-medial inheritance. 49, define pletropy. Give an example. Sickle cell equals disease. One gene affects multiple traits. Example, sickle cell diseases. Slide 50, define ipsis. Give an example. One gene masks another effect. Example, coat color in mice. 51. Define epigenetics. Give an example. Gene expression changes without DNA change. Example, diet affecting gene activity. Define polygenic traits. Give an example. Many genes affect one trait. Example, height or skin color. There's a picture of all the traits and everything. Define extracellular inheritance. Give an example. DNA outside the nucleus traits. Example, mitochondrial DNA from the mom. There's the mitochondrial DNA and the mitochondria. Section 5, central dogma of molecular biology. 54, provide an overview of replication. DNA is copied to pass genetic information during a cell division. Slide 55, define antiparallel nature of DNA. DNA strands run in opposite directions 5, 3, 2, 3, 5, and then you can see the strand in opposite directions. Man, I've gotten fancy with these. 56, describe the function of DNA helicase. Helicase unzips DNA to a double helix, and then that is a picture of a double helix. 57, describe the role of topmere prevents DNA from over-twisting during unzipping, and you can see pictures of that. 58, describe the role of DNA polymerase. Add nucleotides to make a new DNA strand, DNA like replication. And you can see wonderful pictures of that. 59, describe the difference between the leading strand and the lagging strand. The leading is continuous, lagging is one axial fragments. I have all the fragments here. Describe 60. Describe the lagging strand must be read by DNA polymerase backwards. DNA polymerase words backwards on the lagging strip. You can see that all works together. 61. Demonstrate how to replicate a strand of DNA using a base pair match. A with T, T with G, template ATG equals TAC. You can see all the letter combinations over here. Describe the role of DNA telomerase. Extends the telomeres to prevent gene loss and replication. Provide an overview of a transcript. DNA is copied to an RNA, and then you can see all the pictures of the helix, double helix. Oh, look who comes up. It's my kitty cat. 64. RNA polymerase binds protomer on DNA, and then you can see how it all comes together. 65. Describe the initiation and transaction. RNA polymerase builds the mRNA strand. 66. Describe the elongation in the transcript. The RNA polymerase stops and releases the mRNA, and you can see how that kind of separates. 67. Describe the termination in transcript. DNA TAC equals RNA AUG. There's the picture of the gene and the transcription. 67. Demonstrate how a strand of DNA is transcribed to an RNA. Introns equals removed, exons equals kept. Define the difference between introns and extrons. RNA equals general, mRNA equals message copy used for proteins. 70. Describe the difference between RNA and mRNA. Cut out the introns and joins of extrons. 71. Describe what occurs at the spliceosome, protects the mRNA from damage, and helps export. You can see all that going together. 72. Describe the role of polydactyl tail. mRNA builds a protein. You can see the starting point of that translation. Provide an overview of translation. 2 subunits, small and large. Reads mRNA, joins the amino acids, and you can see just kind of all of that around here. 74. Describe the structure of a ribosome, an entry, peptide bond, and EN exit. You can see the peptide bond here. 75. Describe the differences between the A misal site and P site and exit site in the ribosome. Builds proteins each have covered up and unique side chain. Describe the structure and function of amino acids. AUG equals GGU, UAG, UAA equals start, glyce, stop. And you can see all those combinations. 77. Demonstrate how to translate a strand of mRNA to a polypeptide. Start equals AUG, stop equals UAA, UAG, and UAG, UGA. Identify the start codon and 3 stop codons in animals. Ribosome binds an mRNA at stop codon, and you can see the picture over here. Describe the initiation, I can't say it, in translation. The tRNA brings amino acids growing chain, and you can see all the chains going in and out with the peptide system. 80. Describe elongation in translation. Stop codon ends protein synthesis, and you can see the chain over there in the translation. 81. Describe termination transition in translation. Termination happens when a codon signals in the ribosome release 2 peptides to disable the translation machinery, and then you see the translation termination. Now my cat is trying to go under a drawer. Section 6, gene regulation. Describe the enzyme adaptation model of Jacob and Mondio, I don't know. Genes turn on and off in response to the environment. Lagged operation is key example, and you can see that there's all these, there's the operation, how it all works together. 83. Describe the benefits of gene regulation. Saves energy, allows cells to respond to changes and specialize. You can see how they all work together. 84. Describe the transcriptional gene regulation. Controls of RNA is made from DNA. You can see the DNA over there. 85. I got fancy again. Describe the post-transcriptional regulation. Controls how the mRNA is processed, spliced, and degraded. 86. Describe the translational regulation. Describes how often mRNA is translated into proteins. So you can see how that kind of all goes together. 87. Describe an operand. Give an example. Group of genes with one switch equals LSA operation in bacteria, and the lack operation over there. 88. Describe the role of microRNA, and you can see microRNAs are black mRNA or cause its breakdown. And you can see the binding and how that all kind of works together. Section 7, biotechnology. Provide 2 examples of how humans are used biotechnology before the 1990s. Selective breeding and fermentation, bread, wine, yogurt. Define genetic engineering, directly changing the DNA to alter traits of functions. And you can see all that, how that all goes together. Describe the difference between a genetically modified organism and a transgenic organism. GMO, altered genes, transgenic genes from different species, genetically modified organisms, GMO, bacteria, plants, animals, and viruses. Describe the difference between the size complexity of various genomes, bacterial genomes, eukaryotic genomes, viral smallest, bacterial simple, circular, eukaryotic, small complex. 93. Describe the difference between proteomic and geotomic. Geotomic, study of genes, proteomic, study of proteins. That's it.